Hello dear students, this is Dr. Adwet and welcome to lecture number 4 of Cell, the Unit of Life. As always, let's have a quick recap of what we have done in the previous 3 lectures. So, in lecture number 1, we started talking about Latin words and why so many Latin words make an appearance in our subject biology. We saw words like bios, which meant life, logos, which meant study of, auto, which meant self, hetero, which meant different, trophos, which meant to eat, botan, which meant plants, zoon, which meant animals. From here we came to know words like autotrophs and heterotrophs. We also got to know words like biology, which meant the study of life, botany, which meant the study of plants, zoology, which meant the study of animals. And in the first lecture itself, we discussed why is cell called as the unit of life. And I explained to you in detail why is it called unit of life. What does the word unit mean in this context, in the context of a living cell? So this is what we had discussed in lecture number 1. In lecture number 2, we went on to discuss a few more Latin words like micro meaning very small, macro meaning small, mega meaning big, and magnum meaning very big. We also saw that scopion means to see and cyton means cell. From here we came to know that the word microscope would mean an instrument used to see something which is very small. And then we went on to discuss the history of microscopy and we saw what is cytology. Cyto meaning cell, logos meaning study of. So cytology would mean study of cell which requires a microscope, so it would be called as microscopic study of cells. We also saw what is the cell theory and what does the cell theory state. So the cell theory proposed by 3 different scientists says that all living things are made up of cells or products of cells, and the second point being that all new cells come from pre-existing cells. So this was lecture number 2. In the last lecture, we saw things like concept of what is metabolism. So I explained to you that whenever little things or little molecules combine together to form bigger molecules, it is called as anabolism. At the same time, whenever a big molecule will break down into smaller molecules, it is called as catabolism. And these are the only 2 main kind of reactions that are happening inside a living organism. छोटा छोटा लेके जोड़ के कुछ तो बड़ा बनाओ, या तो कुछ बड़ा लेके उसको तोड़ के कुछ तो छोटा छोटा बनाओ. So you have anabolism and you have catabolism, and both these things together are called as metabolism. Anabolism plus catabolism equals to metabolism. So that's the first thing we discussed in lecture number 3, that what is the concept of metabolism. Then we started speaking about parts of a cell, and when we discussed parts of a cell, we saw that cell has a living material which would be called as protoplasm. Now this living material protoplasm in a few cells can be protected by a non-living tough covering which is called as a cell wall. All cells do not have a cell wall. Some cells have a cell wall, some cells do not have a cell wall, but all of them will have the living material called as protoplasm. Plant cells have cell wall, animal cells do not have cell wall. Now this living material, protoplasm, can be further classified into 3 main areas. The first is going to be the controlling center, which will be called as the nucleus. The second is going to be the executive center, which will carry out all the work, which will be called as the cytoplasm. And the third will be the boundary which keeps all of this protected and allows things to go outside, inside, and will manage all of that. That boundary being called as the plasma membrane or the protoplasmic membrane or the cell membrane. Here I would like to again remind you that cell membrane is plasma membrane and cell wall is outside the cell membrane, outside the entire living thing. Cell wall is dead, cell membrane is living. So these are the 3 components of the cell. Now, nucleus contains the genetic material in the form of DNA and RNA that is responsible for the transmission of hereditary characters. The cytoplasm contains non-living things like water and living components like the cell organelles. So this chart which you see represents all the things which are present inside a cell. So that is what we did in the previous lecture. Then we continued with the 2 Hindi anecdotes which I shared with you about my father, Dhirubhai Ambani, which was explaining to you as to what is the whole concept behind the genes and how they operate and how they are stored inside the nucleus. So here I explained to you after those two anecdotes that just like you have a cupboard which is going to store books which are made up of pages on which material is going to be written about what and exactly what, किसके बारे में और क्या लिखा है. Similarly, you are going to have a nucleus which will contain the chromosomes which are going to be made up of DNA, and on that DNA information is going to be written. किसके बारे में लिखा है will be called as genes, exactly क्या लिखा है will be called as alleles. Color of the eye gene है, अगर आप black कहे, brown कहे, blue कहे तो वो alleles हैं. किसके बारे में बात हो रही है? Color of the eyes. Exactly क्या बात हो रही है? Brown है, blue है कि green है. So DNA will comprise of genes, and what is actually written there is going to be called as the alleles. So now you know that you have a cupboard के अंदर books which are made up of pages जिसके ऊपर किसके बारे में क्या लिखा है. Similarly, you have a nucleus. Inside the nucleus you are going to have the chromosomes. The chromosomes will have the DNA, and on the DNA you will have genes, and the specific features or the specific types of genes are going to be called as alleles. Today we will discuss a little bit more about this subsequently in the lecture. So this, what you see here on your screen right now, I hope that everybody is now crystal clear about how this is working, because this is the basic knowledge on which I am going to try and build up entire genetics in the future. So this I hope is extremely clear with everybody. Okay, moving on. After this, we saw what are the characters of a cell or characteristics of the cell. And here we saw that there are 6 characteristics of the cell. The cell is the structural and functional unit of life. The cell is going to be made up of living material which is going to be called as protoplasm, which is surrounded by a protoplasmic membrane or a plasma membrane. Inside that you are going to have a controlling center which will be called as the nucleus, and an executive center which will be called as the cytoplasm. The nucleus is going to be containing the genetic material in the form of DNA and RNA that will be responsible for transmission of hereditary characters. At the same time, the cytoplasm contains the various cell organelles which are going to carry out the metabolic activities like anabolism, catabolism, jodna todna. And lastly, the cell is responsible for division and growth. It can also repair itself. So these were the characteristics of the cell. And then I showed you this image and I asked you, what kind of a cell do you think this is? Majority of you gave the right answer that this is a plant cell based on the cell wall that you were able to see. In fact, in the last lecture, I had also mentioned that cell wall is one of the reasons why plant cells appear to have a fixed shape. They are not dynamic. They appear to have a fixed shape because this dead cell wall doesn't really move or doesn't really help them to change their shape, allow them to change their shape. So everybody correctly said that this is a plant cell. Here, I also showed you this cell, which is a bacterial cell, which is much smaller as compared to a plant cell. And then here I explained to you that plant cells or animal cells have a lot of features, lot of characters which they want to pass on to their offspring. That means they have a lot of characters. If they have a lot of characters, what will they require? Lots of pages. If they require lots of pages, what will they require? Lots of DNA. If they require lots of DNA, that can be made into lots of books like chromosomes. So in this case, in the case of a plant cell, there will always be lots of chromosomes, and to keep these chromosomes, lots of these books, you will require a nucleus, a proper nucleus. So here I explained to you that carrion is a nucleus, U means true, and this is a eukaryotic cell, a cell with a true organized nucleus with the presence of a nuclear membrane. On the other hand, I spoke about this cell, the bacterial cell, which has no cell organelles or no characters as such, at least not as many as the eukaryotic cell. So if there are less characters, that means that there is less information to pass on, which will require less pages, so less DNA. So all the DNA can be compressed into one single chromosome, and for that one single chromosome, you will not make a full cupboard. For that one book, one full cupboard will not be made. So here you don't have anything like a proper nucleus. It is just one single book, one single chromosome floating around in the cytoplasm. So this cell is going to be called as a prokaryotic cell. Pro meaning primitive, karyon meaning nucleus. So this is a prokaryotic cell. So here is where we saw what is the difference between a prokaryotic cell and a eukaryotic cell. And this, dear children, is where we had finished the last lecture. So today's lecture starts now. Before I start today's lecture, I just want to give out a warning to everybody. If you are starting to watch this lecture series at this point, from this lecture, the 4th lecture, please stop and ensure that you have seen the previous 3 lectures. Today's lecture will make very little sense if you have not seen the previous 3 lectures. If there is any doubt regarding the previous 3 lectures, please see it again. If you need any clarity, you know how to contact me. So ensure that the previous 3 lectures are seen, and only then should you be starting at this point. I have told you in the very first lecture, I would like to repeat this again, that if you have a strong foundation, then you can build more knowledge on top of it. So today, whatever we will do will be built upon the knowledge that I have tried to pass on in the previous 3 lectures. So ensure that the previous 3 lectures have been seen, have been understood. If you have any doubts, please get them clarified first and then start off with this. So let's start today's lecture. So now for the next few lectures, I am going to be talking only about eukaryotic cells. We will not discuss prokaryotic cells right now. We will discuss them after we finish entire discussion about eukaryotic cells. So eukaryotic cells have a nucleus, have a cytoplasm and have a plasma membrane. Today I am only going to talk about the nucleus. So today we will finish off the eukaryotic nucleus. Next lecture we will be starting off with the organelles. So for the next few lectures we will be talking about organelles inside a eukaryotic cell, and once we finish that we will talk about plasma membrane of a eukaryotic cell. With that we will finish entire eukaryotic cell, and once those concepts are clearly understood by everybody, we will move on to the prokaryotic cell. So let us start with the nucleus of a eukaryotic cell. So it all begins with this person you see on your screen, whom you've heard about before. He is Robert Brown, and I have briefly mentioned his name when we were talking about Anton von Leeuwenhoek. So he was living in the earlier part of the 19th century, and this is the microscope that he was using. So he was using this microscope and he was examining the leaf of the orchid plant. I'm sure everybody must have heard about orchid flowers. So he was working with the leaf of the orchid plant, and this was the microscope that he was using, as you can see on the screen. So that was his microscope, and he was looking at the upper layer of the leaf. So the upper layer of the leaf is called as the epidermis. So he looked at the upper layer under the microscope, and in 1831 he was able to see this, the picture that you see on the screen right now, this picture. So what he noticed was that the cytoplasm was fairly transparent, but there were these dark opaque areas. Can you see that? Can you see that? And these dark opaque areas were the ones he labeled as the nucleus. So he was the first person to describe that the nucleus exists inside the cell. So if someone was to ask you in NEET examination that nucleus was discovered by, it was Robert Brown. Which plant was he working with? Orchids. So the nucleus was first discovered in a plant cell. It was not discovered in an animal cell. That could also be another form of an MCQ. Okay, let's move ahead. So the next scientist that we have to talk about is this person here, Mr. Walter Fleming, who was living in the later half of the 19th century. His contribution was that he improved upon Robert Brown's technique by using special dyes to color parts of the cell. So he started using these basic alanine dyes, or these basic blue colored dyes. So imagine this. Now you already know that the nucleus is going to contain nucleic acid. You have done this in school. So nucleic acid is acidic. So if you take some blue colored chemical which is basic and you put it on the specimen, what do you think will happen? The acidic nucleus will try to attract the basic color that you put on it. So the minute he started using these kind of chemicals, he was able to see what was inside the nucleus, the nucleic acids, very, very clearly. In fact, this is one of his hand-drawn pictures. So this is what he saw inside the nucleus. So when he saw these things inside the nucleus, he called them as chromatin. So at the point that he saw this in the microscope, we did not even know that there were nucleic acids or things like that. The only thing that he knew was that when he put this color, the center part of the cell, the central part of the cell, was absorbing this color and was looking orangish or bluish in comparison to the rest of the cells. So what he saw in the center, he termed as chromatin. He termed it as chromatin. Chroma meaning color. So the colored substance that he saw inside the cell, he termed it as chromatin. Chromatin, material of the nucleus stained by the basic dyes. So he is credited with discovering chromatin. Now, if you've understood the previous concepts that we have discussed, that the nucleus contains the chromosomes are going to contain DNA, the DNA will have the genes. So the DNA, basically, the deoxyribose nucleic acid, DNA, need to be glued together or to be put together so that they can form a chromosome. To do that, you require proteins. So once these proteins, the glue that holds the chromosome together, plus DNA, the material with which you make chromosomes. So pages called DNA make books called chromosomes. To join these pages to make the book, you will require proteins. So those proteins along with the DNA are called chromatin. You are going to study this in much more detail in the coming chapters when we talk about cell division. So right now I just want you to wrap your head around this, that there are pages called as DNA which make books called as chromosomes. To hold these pages together, you require a glue-like material which you can call as these proteins. And these proteins plus these DNA molecules together form something called as chromatin. Who was the person who discovered this chromatin for the first time? It was Walter Fleming. And Walter Fleming used basic dyes to see this material inside the nucleus. So chromatin, material of the nucleus stained by the basic dyes, was first coined by Walter Fleming, and that was in the year 1882. In fact, Walter Fleming is the same person who also coined the term mitosis. So this is what you need to know about Walter Fleming. Is that clear? So the first scientist was Robert Brown and the second scientist is Walter Fleming. So that brings us to this. Have a look at this cell and think for a couple of seconds as to whether it's a prokaryotic cell or a eukaryotic cell. Alright, I think majority of you must have understood that we can see a lot of cell organelles. We can also see that in the center there appears to be a nucleus. The nucleus is separated from all the cell organelles of the cytoplasm. So I think that everybody now has a fair idea that this is a eukaryotic cell. Now can you think whether it's a eukaryotic plant cell or a eukaryotic animal cell? Are you able to perceive or able to see any kind of a cell wall in this diagram? Correct. So there is no cell wall, which means that this cell is an animal cell. And now we are going to talk about its nucleus. So whatever we speak from henceforth or whatever I explain to you henceforth will be applicable to a plant cell as well as an animal cell, because both of these are eukaryotic cells and they will have a nucleus. So if we take this eukaryotic cell into consideration, you can now see that I have highlighted the nucleus and made the other parts less relevant. So you can see that the nucleus is clearly visible, that organelle which you can see here is the nucleus. If I was to magnify it here, this is what you will see. So this whole picture is this nucleus which has been magnified. Has everybody understood that? Now, eukaryotic cells have at least one nucleus per cell. So the NCERT says that normally there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. So the norm is एक cell एक nucleus, but there are some cells like human RBCs which do not have a nucleus at all. Why is that the case? You will be studying in future chapters. Also, there are some human cells like the muscle cell which have a lot of nuclei. There are some fungi also which have a lot of nuclei. Now, why is that? Is also something you will be doing in future chapters. As of today, I just want you to know that eukaryotic cells have at least one nuclei, but there are variations. There are some cells which may have no nuclei, and there are some cells which may have a lot of nuclei. Let's now focus on this nuclei. As you can see, this nucleus is a well organized nucleus and it has a nuclear membrane. So as should be clearly obvious in this diagram now, you can see that this is a spherical nucleus, it's a sphere, and it has this boundary that I am tracing right now. It has this boundary. This boundary that you can see here is called as the nuclear membrane. Everything within the boundary is the nuclear matrix, which I had also told you last time is called nucleoplasm. So there is a boundary called as the nuclear membrane, and inside it is going to be the nucleoplasm. What is the nucleoplasm going to contain? Now I think you have an idea. The nucleoplasm will contain the chromosomes, will contain the chromatin material. What is the chromatin material? You are going to take the DNA and you are going to combine it with proteins, and what you get is chromatin. So all that chromatin material will be there in the nucleus, in the nucleoplasm, in the nuclear matrix, and there will also be this thing which you see in the center, which is called as the nucleolus, So we are going to discuss each of these stepwise. So broadly speaking, a eukaryotic nucleus has a membrane. That membrane ensures that cytoplasm remains outside and nucleoplasm remains inside. The nucleoplasm can also be called as the nuclear matrix, and the nuclear matrix contains chromatin and it contains nucleolus. Is this clear to everybody now? Okay, let's move ahead. Now, if I was to take this section of the nuclear membrane, can everybody see that? I have just taken this box here. Can you see this white box? We are now going to enlarge it, and I am going to enlarge it slightly more so that everybody can understand this in detail. So here is the nuclear membrane. The nuclear membrane is going to separate the inside part, the nuclear matrix. What is the nuclear matrix made up of? Two things. It will have chromatin and it is going to have the nucleolus. So that inside part will be separated from the outside cytoplasm and the outside cell organelles. So that boundary is going to be called as the nuclear membrane. It can also be called as the nuclear envelope. Now the nuclear envelope or the nuclear membrane, as you can see here, has a number of these holes. In fact, you can see here we have actually cut right through a hole. So you can see that this is the outside part called cytoplasm. This is the inside part called as the nucleoplasm, and things can go from here to here. So the nuclear envelope is the boundary which separates or differentiates the nucleoplasm from the cytoplasm. It will separate the nucleoplasm from the cytoplasm. At the same time, proteins or RNA materials like that need to go from outside to inside or inside to outside. So you have holes in the nucleoplasm which allow that to happen. Those are called nuclear pores. So the nuclear envelope has nuclear pores. At the same time, you can see that this membrane is double layered. So here you see the nuclear pores, and you see that there is a outer nuclear membrane and there is also an inner nuclear membrane. So this boundary of the nucleus is not a single membrane, but it's a double membrane. There is an outer membrane and an inner membrane. What this means is that between the outer membrane and the inner membrane, there will be some space. Now that space is called as the perinuclear space. What is it called? Perinuclear space. The word peri means outside and surrounding. The word peri means outside and surrounding. So nucleus के बाहर और surround करती हुई जो space है उसकी दो membranes के बीच में will be called as the perinuclear space. NCERT also mentions that it is about 10 to 50 nanometers. So what have you understood about the nuclear envelope so far? That there is a nuclear envelope which ensures that अंदर का nuclear matrix या nucleoplasm is differentiated from cytoplasm. The nuclear envelope is going to be double layered with an outer nuclear membrane and an inner nuclear membrane. उसके बीच में जो जगह है will be called as perinuclear space, about 10 to 15 nanometers. And this is going to keep things outside and अंदर का things inside. but to allow material to pass like proteins and RNA, there will be certain holes, and these holes are going to be called as nuclear pores. Is this clear with everybody? Are you sure? Okay. Now in NCERT, there is a line, and that line says, a number of places the nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. Now to understand this point, I am going to show you another diagram. So here you can again see that there is a nucleus with this part being called as the nuclear envelope. You can see कि ये अंदर जो है will be the nucleoplasm, जिसमें ये वाला part will be called chromatin, and this part is going to be called as the nucleolus. Here you can see that this is the outer membrane and this is the inner membrane, and as you can see they are continuous in this part. So ये जो nuclear pores है, you can see all these are nuclear pores. So इधर भी एक nuclear pore था, and जिधर ये nuclear pore है, can you see that these membranes are continuous? So this is the point that I want to stress upon from NCERT. NCERT says that nuclear pores are formed by the fusion of its two membranes. Now this actually is a very, very old concept about nuclear pores. This picture which we have been doing right now, or which I just explained to you, is actually a more recent theory of how nuclear pores look like or operate. In fact, you can see that यहाँ पर भी the outer membrane is continuous with the inner membrane, but जिधर ये pore है, you also have these pore proteins. Pore के यहाँ पे proteins which are also going to play a role कि क्या अंदर आ सकता है या क्या अंदर से बाहर जा सकता है. So this is a more recent explanation of how a nuclear pore looks like. But even in this, the outer membrane is continuous with the inner membrane. Alright. So now let's have a look at the actual electron microscope images. So this is how the nucleus looks under an extremely advanced modern microscope known as the electron microscope. This is how it looks like. And here you can see that ये जो तुमको दिख रहा है, ये this is the outer membrane, and यहाँ पे outer membrane घिस गया, तो what you can see here is probably the inner membrane. So अंदर का membrane, ये जो उसके ऊपर है वो outer membrane, and all these holes or openings which you are seeing here are nothing but the nuclear pores. So if I was to just magnify this much area here, just this much area of the nuclear pores, this is how it appears to be. So this is how we know by modern electron microscopy that the nuclear pores actually have these proteins which are going to protect the pore. So what are the parts of a eukaryotic nucleus? You have the nuclear membrane or the nuclear envelope, and you have the nuclear matrix. We have just finished the discussion on the nuclear membrane or the envelope. So I hope that part is now clear to everybody. So we can now start talking about the nuclear matrix or the nucleoplasm. As I previously explained to you, the nucleoplasm has two things. It has chromatin and it is going to have the nucleolus. Now, what is chromatin? As discussed earlier, chromatin is nothing but DNA combined with proteins which can be stained, which can be colored by using basic dyes. So chromatin is the material of the nucleus stained by the basic dyes. It is basically proteins combined with DNA. Now here I want to talk a little bit about DNA. So what you see on your screen right now is an animal cell, and here what you see is a bacterial cell, also called as a prokaryotic cell. So you have a eukaryotic cell and a prokaryotic cell. Both of them will have DNA. Both of them will have pages on which information will be written. So both of them have DNA. In this case, the DNA will form many books or many chromosomes. Here, the DNA will form only one book or one chromosome. That you already know. Now, I am sure in school you have seen that this is how DNA looks like, or everybody has a fair idea. I don't expect you to know today as to how this structure was decided, इस structure में कौन सी चीज़ क्या है, etc. etc. I just need you to know that DNA looks something like this, and it's a helix, it's a helix, it's a helix. So as long as you know that much, that this is what DNA looks like, it is sufficient for our discussion right now. So both these cells, the prokaryotic cell and the eukaryotic cell, have DNA. So इधर का DNA in the animal cell looks like this. In the prokaryotic cell also, it looks like this only. So is there a difference between prokaryotic DNA and eukaryotic DNA? Now there are a few differences. The major difference is that they both look exactly like this, but the difference being in a prokaryotic cell, the DNA is like this. Now what do I mean by the DNA is like this? In an animal cell, in my cells, the DNA looks like this. And the thing is that you can find the two ends of the DNA. Meaning that this DNA, which will be inside this nucleus, there might be a very long DNA, it might be coiled, etc., etc. But you will be able to see that there are two ends. It starts somewhere and ends somewhere. Such DNA is called linear DNA. Such DNA is called linear DNA. Eukaryotic cells have linear DNA. The next, prokaryotic cells, these ones, they also have DNA which is like this. But as you can see in the image, what happens is that if you were to look inside a prokaryotic DNA or a prokaryotic chromosome, it appears exactly like eukaryotic DNA, but you will not find the two ends. It will start somewhere and you will see here, and then it will come back and join here only. So you will not find the two ends. And because of that, in a prokaryotic cell, the DNA is called as circular DNA. So what is the difference between eukaryotic and prokaryotic DNA? Eukaryotic DNA is going to be DNA which is going to have ends which are basically there present. You can find them if you want to. Whereas in a prokaryotic cell, the DNA structure is the same. It will be the same helical structure, but you cannot find the end because they are joined. So basically it goes round and round in circles, and hence it is called as circular DNA. So you have linear DNA and circular DNA. So that finishes our discussion on chromatin. So the nucleoplasm, nucleoplasm has chromatin, and here it has something called as the nucleolus. So what do you need to know about the nucleolus? For today's discussion, you just need to know that inside the nucleus there is a dense area called as the nucleolus. There is a dense area called as the nucleolus. It does not have a boundary, which means that whatever is there in the nucleolus is continuous with the nucleoplasm. मतलब you cannot figure out कि भाई इधर nucleolus खत्म हुआ और इधर nucleoplasm चालू हुआ. So it appears to be continuous, but it is darker than the surrounding nucleoplasm. It helps the cell to make proteins. So it helps in protein synthesis. So this tells you that if a cell is making a lot of proteins, inside the nucleus, it is going to have a lot of nucleoli. So one single one, as you can see here, is called as a nucleolus. Many of these will be called as nucleoli. Is this concept clear to everybody now? So what do you need to know about nucleolus? They are present in the nucleoplasm. They are spherical bodies. They do not have a membrane or a boundary. The material of the nucleolus is continuous with the nucleoplasm. They help in protein synthesis, and in cells which are actively doing a lot of protein synthesis, you will have multiple nucleoli. Nucleoli being plural and nucleus being singular. So just to have a quick recap of what we have done so far, the nucleus in a eukaryotic cell has a boundary called as a nuclear envelope. It is going to have two layers, an outer layer and an inner layer, between which you have the perinuclear space, which is about 10 to 50 nanometers. There are going to be openings at regular intervals which will allow material from inside to go outside, or material from outside to come inside. They are called as nuclear pores. At the same time, whatever is inside the nuclear envelope will be called as the nuclear matrix or the nucleoplasm, which have two things. They will have the chromatin and the nucleolus. Is everybody clear with chromatin, which is DNA plus proteins, and you are going to have the nucleolus, which is going to help in protein synthesis? Clear? So that brings us to what are the points that you need to know about eukaryotic nucleus. If you are paying attention so far, all of these are now very easy. So a eukaryotic nucleus is a true organized nucleus with the presence of a nuclear membrane. Because that boundary or membrane is there, nucleoplasm, cytoplasm se separated hai. So nucleoplasm is differentiated from cytoplasm. As discussed in the last lecture, it will always have more than one book. So eukaryotic cell will always have more than one book. That is why the nucleus exists. So there will always be more than one chromosome. There will be linear DNA as compared to circular DNA in prokaryotes, and nucleolus will be seen in a eukaryotic nucleus. Nucleolus will be seen in a eukaryotic nucleus. Lastly, we saw the functions last time itself, that nucleus is the controlling center of the cell. It is going to contain the genetic material in the form of DNA, RNA that is responsible for transmission of hereditary characters. Are these points now very clear with everybody? Fantastic. What this means is, today itself we can discuss what is the difference between the nucleus of a prokaryotic cell and a eukaryotic cell. Let's see if you can speak along with me or if you have understood this clearly. A eukaryotic nucleus has a true organized nucleus, whereas a prokaryotic cell does not have a true organized nucleus at all, because there is no boundary, because there are no multiple books, there is just one chromosome. So there is no boundary to keep that chromosome intact. So in the case of a prokaryotic cell, that nucleus jaisa area, not nucleus, that nucleus jaisa area can also be called nucleoid. So here there is a true organized nucleus which you can see here, and here this area, this rough area can be called as a nucleoid. The second thing is that in the case of eukaryotes, a nuclear membrane is present, whereas in prokaryotes, nuclear membrane is absent. In the case of eukaryotes, nucleoplasm is differentiated from cytoplasm, and here there is no boundary only. So जो भी nucleus के आजू बाजू का material है वो cytoplasm के साथ continuous है. There will always be more than one chromosome in a eukaryotic cell, whereas there will always be only one chromosome in a prokaryotic cell. There will be linear DNA and there will be circular DNA. Nucleolus will be there and nucleolus will be absent. So very quickly, what are the differences between a eukaryotic nucleus and a prokaryotic nucleus? True organized nucleus present, true organized nucleus absent. Instead, the rough area in which you can see that one chromosome will be called as the nucleoid. So true organized nucleus present, true organized nucleus is going to be absent. Next thing is going to be nuclear membrane will be present, nuclear membrane will be absent. Nucleoplasm will be differentiated from cytoplasm. Nucleoplasm is continuous with cytoplasm. Will always show more than one chromosome, will only show one chromosome. Will have linear DNA, will have circular DNA. Nucleolus will be present, nucleolus will be absent. Is everybody absolutely crystal clear about these points? Fantastic. So with that, let's see what NCERT has to say about the nucleus. So I'm going to read through this because these are points which we have completely covered while talking about the nucleus today. So we saw that nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming, Walter Fleming. This was somewhere around 1882. NCERT has not mentioned the date. Normally, there is only one nucleus per cell in most of the cells. Variations in the number of nuclei are also frequently observed, as in the case of RBCs where there is no nucleus at all, or in the case of muscle cells where there are multiple nuclei. The interphasic nucleus, the nucleus of a cell when it is not dividing. So as of right now, just remember that the interphasic nucleus means the nucleus of a cell which is not dividing. Don't need to focus on that now, just focus on the term nucleus. So the nucleus has a nuclear envelope and it also has a nuclear matrix, also called nucleoplasm. And the nuclear matrix or the nucleoplasm contains chromatin, which is highly extended and elaborate nucleoprotein fibers. So basically nucleoprotein meaning DNA and proteins. So nucleoprotein fibers, and it contains nucleoli, which I explained to you are those bodies which help in protein synthesis. Plural is nucleoli, singular is nucleolus. So let's talk about nuclear envelope first. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space in between which is about 10 to 15 nanometers, called as the perinuclear space. So outer nuclear membrane, inner nuclear membrane, and then the perinuclear space which is about 10 to 15 nanometers. It forms a barrier between the materials present in the nucleus and that of the cytoplasm. So nucleus के अंदर का अंदर रखो, nucleus के बाहर का बाहर रखो. Nucleoplasm और cytoplasm को separate करने के लिए you have a nuclear envelope, a double layered nuclear envelope. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes, which hopefully now you've clearly understood from the diagrams that we have seen. These nuclear pores are the passages through which movement of material like protein molecules takes place in both directions, meaning that material can go from nucleoplasm to cytoplasm and material can also go from cytoplasm to nucleoplasm. Lastly, the nuclear matrix has two things. It has chromatin and nucleolus. The nuclear matrix or the nucleoplasm contains nucleolus and chromatin. Chromatin, highly extended and elaborate nucleoprotein fibers that can be stained by basic dyes, first observed by Fleming in 1882. And the nucleoli, they are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane bound structure, does not have a membrane. That is why uske jo bhi contents hai are continuous with the nucleoplasm. It helps in protein synthesis, and larger and more numerous nucleoli, more than one nucleoli, are going to be present in cells that are actively carrying out cell division. So this is what the NCERT says about the nucleus. So I hope that the nucleus as a cell organelle is now extremely clear with everybody. So as far as a eukaryotic cell is concerned, it is going to have protoplasm and it is going to have cell wall. The protoplasm comprises of the nucleus, the cytoplasm and the plasma membrane. We have finished the nucleus today, and when we meet again next time, we are going to be talking about the organelles. And that, dear students, concludes today's lecture. I hope you have understood everything, and I cannot wait to see your smiling faces in the next lecture. Thank you. God bless. Work hard. Be nice.